Well, good morning, and thank you everyone for joining us today. Queriah Simpson is going to be defending her thesis as a requirement for her Master of Science degree in Environmental Science at FAMU with a concentration in marine and estuarine environments. She is a NOAA Center for Coastal and Marine Ecosystems graduate scholar. She previously earned her Bachelor of Science degrees at FAMU in Environmental Science and also in pre-professional biology with a chemistry minor. She was a scholar with the NOAA Environmental Cooperative Science Center. And welcome, Queriah.
Thank you, Dr. Maury. Good morning, everyone that's online. I appreciate you joining to listen to my research. Again, my name is Queriah Simpson, a NOAA CCME graduate scholar at Florida A&M University. I will be defending my thesis work, the application of habitat suitability models for benthic communities in the Eastern Gulf of Mexico, linking bioprospecting and modeling research. First, I would like to thank Florida A&M University School of the Environment and NOAA Center for Coastal and Marine Ecosystems for granting me the opportunity to conduct this research. Thank you to my committee, Dr. Stephen Maury, Dr. Bernadette Kelly, Dr. Michael Abazingi, and Dr. Sharmani Pitter. I would also like to thank my mentors at NOAA's National Center for Coastal Ocean Science, Biogeography Branch in Silver Spring, Maryland, Dr. Holly Goyard, Dr. Arliss Winship, and Mr. Matt Pody. I would also like to give a big thank you to Dr. Kellogg, who works with the United States Geological Survey at University of South Florida. And lastly, but not least, I would like to thank Dr. Sandra Brooke and the science team at FSU for facilitating my participation on the HydroSMAC mission. I will first introduce my topic, then move on to details regarding this research, discuss results, and lastly, introduce the idea for using this research to aid in bioprospecting. Last summer, I had the opportunity to participate in NOAA's Experiential Research and Training Operation, also known as NERDO, at NCOS, the National Center for Coastal Ocean Science Biogeography Branch in Silver Spring, Maryland, where I had the opportunity to assist with the compilation of models that predict areas along the West Florida escarpment that are suitable for deep sea coral habitation. A couple months after my NERDO, I was able to embark on a research cruise called the Hydrodynamics and Habitat Suitability of Meiofauna and Corals, aka HydroSMAC, in which the models I helped produce were used to help identify dive locations that were to be explored during the cruise. This was all a part of a larger NOAA Office of Ocean Exploration and Research funded project titled Combining Habitat Suitability and Physical Oceanography Models for the Targeted Discovery of New Benthic Communities along the West Florida Slope. Dr. Sandra Brooke is the PI of this project, and Dr. Maury was the co-PI for this project. This thesis will present a framework for the application of some new methodologies for habitat suitability models when predicting locations where deep sea coral communities could potentially occur. This is important because there are bioprospecting efforts that could occur in these same areas as deep sea corals and sponges host microbes that have bioactive compounds of biopharmaceutical interest. Due to unforeseen circumstances, the second portion of my thesis was unable to take place. I plan to do some pilot research in which I would have been able to culture some samples from the cruise and isolate specific strands of bacteria that have the potential to destroy other bacteria that are known human pathogens or inhibit the growth of those pathogens. Deep sea coral are similar to shallow water coral or tropical coral when it comes to biodiversity, species variation, size and shape variation, and even colors. However, they're found in the cold, aphotic zones of the deep ocean. They are azooxanthellate, meaning they lack the symbiotic relationship with zooxanthellae commonly found in tropical coral. They rely on surface primary productivity as they feed on organic material that descends through the water column to the seafloor. They also consume small microscopic plankton. Deep sea coral take on numerous shapes like shallow reef corals, and they also grow together on a common skeleton, forming a complex structure that is able to house numerous organisms. The picture on the left shows an octocoral and a stony coral, both inhabited by brittle stars. The picture in the upper right-hand corner shows a black coral inhabited by a squat lobster. These photos are from previous cruises that helped lead to the research efforts I will be discussing in this thesis. Though found at such great depths, deep sea coral is still affected by anthropogenic activities and susceptible to events such as ocean acidification. Some deep sea coral habitats have been listed as areas for habitat areas of particular concern, as they host diverse populations of fishes, making them essential fish habitats. Being that this research was explorative, the areas located during the HydroSMAC mission can be considered newly discovered, and hopefully the information obtained from the cruise will be used to inform management decisions for the benthic communities along the West Florida slope and the West Florida escarpment. Scientists like Dr. Sandra Brook have prior to recent surveys, deep sea coral habitats were thought to be less extensive in the Gulf of Mexico compared to the Atlantic and Pacific. So until now, scientists like Sandra Brook decided to test the theory and have discovered just how abundant deep sea coral communities are in the Gulf of Mexico. Recent surveys have documented the occurrence of deep sea coral communities along the West Florida slope above 1,000 meters, which led them to believe that these same communities or similar communities could exist at depths greater than 1,000 meters. The West Florida slope is located in the eastern Gulf of Mexico between 250 and 350 kilometers off the coast of Florida. It stretches for about 900 kilometers. It is a steep wall dressed with protruding hard rocky substrata, terraces, and other formations suitable for the attachment of sessile organisms. To your left is a small map of the area in which we study. The next slide will be a larger map. It has not been well studied and has become of great interest for benthic research due to the morphology of the escarpment. So here is essentially the study area. This is the West Florida escarpment. This is a picture of a densely populated piece of rock, basically attached to the West Florida escarpment that hosts a very diverse population of deep sea coral. So how are these areas located? By usage of habitat suitability models. Habitat suitability models are empirical statistical models that predict the occurrence of a species in a particular area. It is a cost-effective and non-invasive way to predict the distribution and magnitude of suitable habitat for benthic organisms such as deep sea coral. This study used habitat suitability models to predict areas along the West Florida escarpment that harbor deep sea coral based upon readily available data and other environmental variables using two novel advancements: the usage of absence data and the inclusion of current speed data at 100-meter resolution, which is more accurate than the previously used 370-meter resolution. Data used to produce these models come from numerous sources, such as the World Ocean Database that you see on your right-hand side of the screen. These databases contain information from previous surveys, environmental data such as salinities and temperatures, nutrients, and things that you would need to upload into your R software or your R workspace to complete a model. So One in particular database that I used was the InCost server. They have their own private databases with survey data from surveys from years ago that they compile and keep over the years for this reason and others. But in my case, specifically to use models to predict deep sea coral occurrence on the West Florida escarpment. Here's another example of a database that can be used to identify areas where deep sea coral have already been found along the West Florida slope and escarpment. It also has other areas of the ocean, but since I'm discussing the West Florida escarpment, I decided to zoom in and give you a clear picture to show you that the points that you see here are of deep sea coral that are above 1,000 meters. So far, I believe there were only 10 instances where there were deep sea coral documentation below 1,000 meters, but I'm not sure that that is a part of this map. I used R software to help produce preliminary models. Data files in proper packages were loaded into R. Scripts were then manipulated to incorporate environmental parameters for the models. That sounds pretty simple, but when I first got there, honestly, it didn't take too long for me to figure it out, but it's definitely something that you have to put effort into to understand how to properly code and get your predictors to work correctly to produce the image that you want. Regardless, if everything is coded properly, there will be a map produced in the right-hand lower corner here. I put up this picture so that you can kind of see what RStudio looks like. And you'll write your code here, you'll edit your script here in the code editor box. And the second box is when you run the code, it shows you if everything runs correctly, which is called the R console. The third box shows the workspace and history, which is an area where anything that you've loaded up here, anything that you've done in RStudio in your workspace would be here in the third box. And you can go back and look at different variables that you've added to your R workspace. And lastly, the fourth screen, your plots and files, is where your model or your map will be generated. Models produced for this research were run at 100-meter resolution, which is finer than the previously used 370-meter resolution. These models incorporate absence data and a 10-year ocean circulation model of bottom current speeds near the West Florida escarpment, given to us by Dr. Stephen Morey. 3 models were generated. One included available data on the family of coral called Isididae, commonly known as bamboo coral, another for Stichopathes, which is a genus of coral that belongs to the family Antipathes, which is the third group of coral that we use to test models with. And the Antipathes are known as black coral. Models are tested for accuracy a number of ways. For example, this Isididae pairs plot was generated to test the influence of our predictors. The pairs plot generates correlation coefficients between each predictor variable. The correlation coefficient here between bathymetry and temperature is 0.8, meaning that they are highly correlated, which makes sense due to the fact that temperature generally decreases with depth. And we are looking at aphotic zones in the deep ocean. And the deep ocean is colder than the surface ocean or surface waters of the ocean due to the fact that the surface is influenced by the sun. Two other highly correlated predictors on this pairs plot are slope and roughness, which both refer to the wall characteristics, which greatly influence whether or not deep sea coral have the ability to inhabit the area. So here we have slope and we have roughness, and you have a 0.7 correlation coefficient. Below that, you see speed is also influenced by slope and roughness of the West Florida escarpment, the wall itself. My main goal for this mean bottom current speed was to extrapolate data onto this model. This speed current map was generated by, again, Dr. Moore. He's a physical oceanographer. And with the help of my mentors, the speed current model was loaded into R. As you can see, current speed increases from blue to red. This model was included to see whether or not current speed influences deep sea coral habitation along the West Florida escarpment. Being that the research is currently still ongoing, it is unknown whether or not current speed influences the attachment of sessile organisms along the West Florida escarpment. Each of the three maps produced encompasses the mean current speed model. The points indicate areas that are already known to house the family or genus of coral specified. The points come from data files that were loaded into R that included information from previous surveys. Probability increases from blue to red, indicating that areas that have the potential to host deep sea coral. Here you see the map. I'm using my pointer to locate the points on the map or the model, just to show you that these points are or pertain to data that was already readily available before we went on the cruise. So these points are areas that are above 1,000 meters. They were just used to predict the areas along the slope due to the conditions and the data from the files, to help us predict areas where we may find deep sea coral below 1,000 meters. There seems to be somewhat of a small error in this model due to the fact that there is red in the middle of the Gulf. That can happen. There can be some errors that happen in modeling. However, I really don't remember exactly which error caused the red to be here, but it did give us the points that we needed along the West Florida slope to predict areas along the West Florida escarpment that harbor deep sea coral. Some of these points are also stacked, so there are more points than you see here, but they're kind of on top of each other, which could happen because some of the same areas may be surveyed multiple times. So you'll have multiple accounts of data for that same general area. This is the Stichopathes model. I'm sorry, the previous slide showed the Isididae model, or the predicted areas of bamboo coral. This shows the predicted areas for Stichopathes. The points, again, show areas that Stichopathes have been documented throughout the years from surveys. The different amount of points on these maps could be due to the amount of available observation data as well. So there may be less points on one map and more on another, but that just could be due to more information on a particular family or species of coral more so than the other. This is the Antipathes map. Same general idea up here. You see a lot more points here, so there's a lot more data on the slope, the West Florida slope, pertaining to Antipathes than it is further out, closer to the escarpment at lower depths. These models were further fine-tuned by my mentors at NCOS. They also supplied more bathymetric maps to further detail the morphology of the West Florida escarpment prior to the HydroSMAC cruise. The HydroSMAC cruise can be viewed as a ground truthing effort for the modeling research, a way to test model accuracy, if you will. The models were able to help select ROV dive locations, which had the ability to be adjusted during the cruise using real-time environmental data. That is shown on this next slide. But first, this is the RV Pointer, owned by University of Mississippi. This is the ship I traveled on for about 2 weeks. This is the Global Explorer, which is a remotely operated vehicle that we use to collect samples throughout the duration of our cruise. So using the model, like I said, being that the weather can be unpredictable and you're out in the environment, sometimes you cannot get an ROV dive done due to could be the waves, or you might have way larger swells in the water than you need. And you can't drop the ROV due to the fact that it may hit the boat, or it could hit the escarpment itself. Here are some pictures of me aboard the cruise helping deploy and retrieve the ROV. I know at least 3 days of dives. We were set to do 9 dives. We were only able to do 6. 3 days of dives we had to cancel. 2 due to weather conditions, the other because the manipulator arm on the ROV that's used to collect samples was broken, or not functioning properly rather than broken. Here we have another map. This map here on the right-hand side indicates dive locations and target sites. The target sites were the planned dive targets, some of which we could not deploy the ROV in. Others we were, such as dive 2 and dive 3. And then dive 5 was also done in one of our original target sites. Other than that, there are a few dives that took place that were not at target sites, such as dive 6 and dive 4. This chart here indicates what type of gear was used to collect samples, when the samples were collected, the area in which the samples were collected, and at what depths. Once we got back aboard once we got the ROV back on the ship, we had to remove samples that were collected by the ROV and store them for later analysis. So I helped out with storage and sample collection for some of FSU's further work. Here's some ROV images that were collected during the cruise. On October 4th, we did not collect any samples at all, but we did get some ROV footage. Here, I believe, is an Antipathes on the right. But this slide is just show you the diversity at such depths, even when it's so dark. This left-hand corner right here is a sponge. That's not even a coral, but in general, you see sponges in deep sea coral habitats. This chart states that on October 3rd, 10 octocoral samples were collected, as compared to the next day where we really didn't collect any samples at all. So that goes to show you that when you're out in the field and doing field research, it's somewhat unpredictable, but thankfully we got enough samples. For your model results, the HydroSMAC mission resulted in the discovery of extensive deep-sea coral habitats along the West Florida escarpment. Therefore, the models did indeed predict areas along the West Florida escarpment that are suitable for the attachment of deep-sea coral. Samples are still being analyzed, and upon completion, they will be identified. So some of these samples that we collected were potentially new species of coral. So I'm excited to see what's to come once Dr. Brooke and the rest of the team at FSU finish genomic analysis. The new information collected during the study, though, will contribute to data concerning benthic communities. And that's what we need, because for proper management to occur, you must have as much information as you can about these communities without affecting them greatly. So habitat suitability modeling is a great way to do so. This is a schematic I created to summarize the main components of this research. You start out building your habitat suitability models based upon readily available data. You go out and you ground truth or survey, and you collect samples, you collect footage and a multitude of in situ environmental data. And you perform analyses, laboratory analyses on the ship, as well as when you dock from the cruise and get back to your normal laboratory setting. Now we'll begin to discuss the broad approach or future work, which is using habitat suitability models to aid in bioprospecting efforts. As we are in what now has been termed a post-antibiotic era, it is vital that we find ways to combat multi-drug resistance. Recently, researchers have begun exploring extreme environments such as the deep sea in search of microbes harboring compounds with potential biopharmaceutical use. In a study conducted by Zhu et al., there was a strand of bacteria identified entitled M864. My apologies. It's on the next slide, just in case you couldn't understand me. But M864 was collected from a sponge from the family Oceanoptidae, collected from the Bahamas at about 850 meters below the surface. It produced potent activity against C. diff. C. diff is a bacterium that causes diarrhea and colitis, or swelling of the colon. It is very common in nursing homes, to be honest, and it can lead to death. Being that a possible cure or treatment can be made using a microbe from the deep ocean, there's a need for more deep sea bioprospecting research to be done. The diagram to the left is a summary of the processes involved in bioprospecting. Prior to a research cruise for bioprospecting, habitat suitability models can be used to locate areas of particular concern or interest, as they predict locations containing benthic communities that harbor organisms like corals and sponges that are known to host microbes with metabolites that could potentially be used for biopharmaceutical research. This could lead to more time-efficient and cost-effective deep-sea bioprospecting. During my training with Dr. Kellogg, she explained to me the importance of proper sample collection when aboard a ship. Dr. Kellogg is a coral microbiologist with the United States Geological Survey. She walked me through very important techniques needed when collecting samples aboard a ship, being that samples can be easily contaminated. Quivers, or the tubes in which we collect all of the samples when the ROV is in the water, and all materials that come in contact with whatever samples you have must be sanitized. Other details, such as filling the quivers with fresh water and closing them prior to ROV descent, is also important to make sure that water from the water column is not collected by the quivers. Once the fresh water is expelled, once the cap opens to collect samples in situ, below 1,000 meters or wherever you're collecting, the quiver will now fill with the surrounding seawater, and your sample will still be technically in its natural seawater. However, upon ascent back to the surface, the temperature increases. Therefore, you may lose microbes due to temperature changes. So it is imperative that you move quickly once the ROV is back aboard the ship to grab the quivers and collect the samples to reduce A, loss of microbial species that could be important, and B, reduce contamination. The materials that you use to handle the samples must also be sterilized before and in between and after sampling to, again, attempt to prevent contamination. The samples must be crushed down into what Dr. Kellogg called a slurry, and that is inoculated on top of a semi-solid agar or selective media that is used to weed out the species of microbes that you do not need. So you're going to lose those weedy species, and you're going to end up with the microbial species that you're targeting. For example, if you're looking for actinobacteria, you would find a selective media that is able to feed and allow actinobacteria to grow. Once you get back to the lab, the cultured samples should be isolated in order to find the bacterial strands of interest. Those same strands are then generally tested against harmful pathogens to see if they can inhibit their growth. The strands that show inhibitory activity are then sequenced via a variety of genomic analysis. The structures are also looked at via NMR, different types of spectroscopy. And then you will have identified the microbe or bioactive compound of interest that does the job that you need it to do and attacks whatever pathogen that you are testing it against. All in all, habitat suitability models and bioprospecting research have the potential to coincide and further contribute to the knowledge base on benthic communities. This is another schematic that now includes habitat suitability modeling, the surveying, the bioprospecting portion of the survey, the analysis, and the biotech work. I feel that the field or deep sea research, different bioprospecting research, due to the fact that everything that you find out, you can use to contribute to the accuracy of your models. Also, to give you more information on benthic communities that may need to be protected and also could be used for other resources. Thank you for listening.
Thank you so much, Kira. We'll now move into the question and answer phase of Kira's presentation. If we have questions, please use the hand raise function if you can figure that out. And just scroll over to the right side of your screen on the participant list or use the hand raise button. If you can't make that work, just unmute and ask a question. So do we have any questions for Kira?
This is Dr. Kelly Howard. You're make some recommendations for future works as far as bioprospecting. So your involvement in this particular research, how has that fueled your thought for further research in doing bioprospecting? Could you make that a little clearer for us?
Okay. Being that I wasn't able to do my initial pilot research, it's a little bit more difficult to really give you everything that I want to tell you. But being on the cruise and going through this process, I found out that you could leverage such deep sea research to collect samples that you can do microbial analysis on. So the same coral samples that were collected and sediment samples and sponge samples that we collected out in October, I could use to, I could culture bacteria from those samples that are in or on the deep sea coral and take it back to the laboratory and try to isolate strands that I find interesting, then test those strands against pathogens that are harmful to humans. A clear shot or a clear way of thinking of this is habitat suitability models lead to areas that are of interest of people that do bioprospecting research. So it would be easy to collaborate with, or I believe it should be a little easier to collaborate with a science group doing deep sea research as a way of, I guess, mitigating costs and time. You can hop aboard the same research vessels, possibly, to, piggyback off of the same samples, collect your own samples during cruise research efforts, survey efforts, if that answers your question.
Yes,
Ms. Simpson.
Because the bio.
Ms. Simpson,
Yes, sir.
how are you?
I'm pretty good. How are you?
I'm good. Let me get your thoughts on this. Being that this is NOAA funded activity, and looking at the direction of the bioprospecting part of the research, could you probably see additional work that may be towards looking at microbes that could potentially be suitable for cleanup of our oceans, cleanup of our waters, because there are emerging contaminants that NOAA has been looking at. And some of these are some of the same pharmaceuticals that are getting into our water bottles, for which we don't have a good cleanup for them. We even have nanoparticles that are getting into the water these days. And of course, we have plastics and other things in the waterways that are more geared towards the mission of NOAA, healthy oceans. How would you, in your recommendations, outside of pharmaceuticals, will this, I think, should be a component as we look forward to using deep sea corals resources to help mitigate some of these water quality issues.
Glad you as you're doing research, have, if you have a microbiologist aboard, they can go ahead and do the culturing and isolation, but instead of testing it against harmful pathogens, you can test these microbes against contaminants such as heavy metals, nanoparticles, as you said, that could potentially break them down. In fact, based on research done with, I want to say the Deepwater Horizon oil spill that I looked up, I found out that there's a class of microbes called hydrocarbonoclastics that actually break down hydrocarbons. So those are found in the water column somewhere. So I'm sure that there is a possibility of these microbes that could exist in benthic communities and or on coral especially due to the fact that some deep sea habitats have been affected by oil spills. Something down there is degrading that oil. So I feel as though there are microbes in that environment that do indeed break down contaminants, PAHs, and hydrocarbons that you find and different pollutants that find their way into the ocean.
So, so then I'm asking, are you going to have, at least I've looked at your results, discussion and conclusions and future research, and I'm just wondering, being that this might be of interest to our funding agency, should there be some sense of what your thoughts are, having done this work, to say to the funding agency, this might be an area for which additional work may be needed to find some of the answers to problems that are currently happening in our waters?
Yes, I feel it's the environmentally friendly way of removing chemicals from the water. So if there is any interest in looking at the deep sea to aid in contaminant and pollution cleanup, I would definitely recommend it. And I feel as though it would be a great expansion to NOAA's mission and NOAA's values.
Okay. I'm going to yield to other folks that may have questions because we're going to talk with you later. So I'm yielding. The audience that is on that want to have that have specific questions, it's open.
I think Dr.
Hi, Queriah.
Dr. Abazingi.
Oh,
Will, go ahead, please.
oh, hi, Queriah. Congratulations on everything. Great presentation. So
Thank you.
well worth it. What are you looking forward to after this opportunity?
After this opportunity, as far as like, you want to be a little bit more specific for me?
Well, after this master's program, what are you looking forward to in environmental sciences and policy?
I plan to go on into an MD PhD program just to study molecular science, molecular biology, and take what I've learned thus far from my master's program with me to med school, because we often lose sight of the environmental aspect of human health when you do get into medicine. You more so look at fixing the problem rather than preventing it or finding the source of the problem. So what I've been taught in environmental science, to be honest, is that a lot of health problems are caused by environmental factors. And if we were to pay more attention to the environment, such as the chemicals that we release into the streets and the things that we do that pollute the air, that we could possibly mitigate a lot of the health issues that we have today.
Well said. Excellent. Thank you.
You welcome.
Thank you, Will. Do we have any other questions? All right. Well, thank you, everybody. And thank you, Queriah. You did a great job. That concludes the presentation and question and answer portion of Queriah's defense. The committee will now convene in a different session for the closed portion of the defense. So I would ask Queriah and the committee, please use the other link that I sent out this morning for the closed portion of the defense. And we'll just move to that. Again, thank you so much, everybody. And Matt Potty, I see you are tuned in. Thank you very much for tuning in from the NOAA side of things. And Jenna, thank you so much for tuning in and part of one of our advisors to CCME. And thank you to the many students that came as well. All right.
Thank you.